students, Assalamu alaikum to all of you. Our today's topic is anatomy of leaf. Anatomy of leaf. Dicot and monocot. Dicot and monocot leaf. First of all, when we talk of dicot leaf, we can say this dicot leaf is dorsiventral. It is dorsiventral. Dorsiventral means the dorsal and ventral surface will vary. Maybe it can vary in number of stomata and or in different surfaces. In leaves there is stomata, but this stomata is must for exchange of gases because food formation takes place in leaves. First of all, when we draw the structure here, this is epidermis and this is lower epidermis. This is upper epidermis. First of all, this is epidermis, like this. And you know that this epidermis is single layer. Epidermis upper, so this epidermis is called upper epidermis, and this one we call it as lower epidermis, like this. And I have already told you epidermis is having a single layer of cells, so it is single layered, and also it is outermost layer. This upper epidermis is at this surface is called as adaxial surface. Adaxial surface. And this lower surface of lower epidermis is called as adaxial surface. Adaxial surface. Then there are tube-like structures like this. Tube-like structures. There are tube-like structures, and these like this. There are tube-like structures, and these tube-like structures are elongated, and these are called mesophyll cells. So these are mesophyll cells, but these are called palisade mesophyll cells. Palisade mesophyll. Then there are cells which vary in structure, like this, like this, like this, and these cells are called spongy mesophyll. Spongy mesophyll. Now there is vascular bundle, like this. These are elongated structures. There are mesophyll cells, again mesophyll cells, and again these mesophyll cells are without intercellular spaces. Mesophyll cells, so we will write here, the mesophyll cells are without intercellular spaces. Now after that, there are vascular bundles. There is vascular bundle, and this vascular bundle is surrounded by type of cells like this, like this, like this, like this, and these cells which surround the vascular bundle, these are called bundle sheath cells. For example, it is vascular bundle, and these cells are called bundle. Now there are vascular bundles, and this vascular bundle is surrounded by bundle sheath cells. Surrounded by bundle sheath cells. And what is the function of these bundle sheath cells? These provide protection. To the vascular bundles. Now it was all about the dicot leaf, and we know that this upper surface of epidermis is called abaxial surface, and lower surface of epidermis is called upper surface of epidermis is called adaxial surface, and lower surface of epidermis is called abaxial surface. And the cells present in between upper and lower epidermis, I have told you these are mesophyll cells, and mesophyll cells are of two types in case of dorsiventral leaf or dicot leaves. Mesophyll cells contain also chlorophyll. These contain chlorophyll. These are of two types. First one is palisade mesophyll, and second one is spongy mesophyll. These contain spongy mesophyll also contain chlorophyll or chloroplast, and these absorb sunlight. These cells are having cavities between them. Then I have told you vascular bundles are surrounded by some cells which are called bundle sheath cells. Then bundle sheath gives protection to the vascular bundles, and vascular bundles in dicot leaves vary on the basis of size of veins or veinlets. And for size of veins or veinlets, for example, if this is a leaf, for example, size of vascular bundles vary on the basis of size of vascular bundles vary on the basis of size of venous or midrib, or you can say size of midrib, or and veinlets. For example, this is a leaf like this, and this is middle vein. You can say middle vein or midrib, and there are veinlets. These are veinlets. And these, if this is a leaf, the vein is flat. For example, this vein is flat. and these, it is flat, these winglets are having small. So the size of vascular bundles will be larger. Size of vascular bundles will be larger, but the size of vascular bundles in winglets will be small. Size of vascular bundles in winglets is small. Then we know mesophyll cells or specialized cells which are found in a dicot leaf, which I have told you. Now there is an opening on the surface of there is an opening, as I have told you. There is an opening on the lower surface of epidermis. That means abaxial surface. For example, it is abaxial surface, lower epidermis. There is an opening which we call as stoma. And where this opening is present, it is present on the lower region of epidermis. So exchange of gases occurs through this opening. Exchange of gases occurs through this opening. Now an abaxial surface is on the lower region of leaf. So this contains this lower region, that means abaxial region. It contains stomata, but it contains abaxial surface on the lower region of the leaf contains more number of stomata. It contains more number of stomata as compared to adaxial surface, which is on the upper epidermis. Then we can write features of the dicot leaf. There are features of the dicot leaf. At number 1, we can say abaxial surface has more number of stomata. As I have told you, abaxial surface has more number of stomata. Then there is, I have already told you, there is mesophyll is differentiated. Mesophyll is differentiated into palisade and spongy mesophyll. Spongy mesophyll. Then there is third, that there is an opening on the then there are bundle sheath cells. Bundle sheath cells are present, and I have told you this vascular bundle is surrounded by bundle sheath cells and it gives protection to the vascular bundle. What is its function? To provide protection to the vascular bundles. bundles. Then there is another feature that arrangement of vein and veinlets is reticulate. There is reticulate venation. Reticulate venation. That means arrangement of veins and veinlets. So there is reticulate arrangement there is reticulate venation. It means that arrangement of veins and veinlets is reticulate. Then there is stomata. Stomata is bean shaped. Stomata is bean shaped. Stomata is bean shaped in case of dicot leaf. For example, like this. It is bean shaped stomata. Bean shaped stomata. Now we can write here what changes we observe between dicot leaf and monocot leaf. So we will take monocot leaf. Monocot leaf. This certain changes which are observed in monocot leaf, we can say changes observed in monocot leaf. Changes which we observed in monocot leaf. First of all, we will see this is isobilateral. Isobilateral. It means that both adaxial and abaxial surface have same number of stomata. For example, both adaxial and abaxial surface have same number of stomata. And then there is another feature of monocot leaf. That means that epidermis is same. Epidermis is same. Then there is third feature that mesophyll cells are not differentiated. Third is mesophyll cells are not differentiated. In case of dicot leaf, these mesophyll cells were differentiated into spongy mesophyll and palisade mesophyll. But in case of monocot leaf, these mesophyll cells are not differentiated. Now there is another feature that cells of the adaxial surface get modified. Cells on the adaxial surface get modified. Adaxial surface get modified. So what type of modification? These cells become larger. Cells become larger, empty, larger, empty, and colorless. And colorless. Now these specialized structures are called bulliform cells, colorless. So these specialized structures are called bulliform cells. Bulliform cells. For example, in case of gross layer. In case of gross layer. So what is the function of these bulliform cells? These cells curl. These cells or curl the leaf and form upper surface. So these cells curl the leaf from upper surface. That is, they help and these also help to reduce the loss of water. These cells help to reduce the loss of water. And there is venation. Veins and veinlets arrangement is parallel. Veins and here is parallel venation in case of monocot leaf. Then parallel venation. Parallel. So we can write here. In case of monocot, there is venation is parallel. But in case of dicot, we have seen there was reticulate venation. But in case of this monocot, there is parallel venation. Now then there is another feature, that size of vascular bundles varies on the size of veins and veinlets. Size of vascular bundles varies on the basis of veins and veinlets. Now there is, I have shown you the leaf that midrib is large, so vascular bundles will be large. When midrib is large, midrib, or you can say vein, when midrib is large, vascular bundles are also large. Now I had told you these mesophyll cells are not differentiated into palisade and spongy. So in case of monocot stem, we can say, or we can write here, mesophyll cells are undifferentiated. Mesophyll cells are undifferentiated. For example, I will draw here the structure of monocot leaf. At last, I will draw here the structure. For example, this is epidermis, upper epidermis, and this is lower epidermis. And I have told you about epidermis, that this epidermis is single layered epidermis, and this is epidermis, upper epidermis, and as such was in case of dicot leaf, upper and lower epidermis, it is lower epidermis. Then this is abaxial surface, and this is adaxial surface. So there are some cells which we call as butiform cells. There are cells like this, which we call as butiform cells. Then there are vascular bundles. Vascular bundles, as I have told you, their size depends upon the size of veins and veinlets. So these are varying vascular bundles. Vascular bundles which vary in shape. Then there are some cells which are mesophyll cells, but these mesophyll cells are non-differentiated. There are mesophyll cells, but these mesophyll cells, or we can say these are undifferentiated. Now, it was all about your monocot leaf. Thank you very much.